Hi guys, this video is about the cell cycle. So the cell cycle is cell division. So you learned in 9th grade biology about mitosis and meiosis, different forms of the cell going from one cell to two. So when it splits, it duplicates its chromosomes, its DNA, all of that. We're going to learn about it in a little bit more detail in here. So the purpose of the cell cycle and cell division is growth and development. So for example, going from a fetus to a baby to an adult, there's lots of growth and development that needs to happen. You need more and more cells. For those cells to be generated, the cell cycle has to happen. Mitosis has to occur. So tissue repair and renewal also. So if you get a cut, your cells have to do mitosis in the cell cycle to generate more. Or just as you live and as you grow, your cells have to renew themselves because they don't last forever. Also, reproduction. So single-celled organisms can reproduce using the cell cycle and mitosis. This is referred to as binary fission. So if it's a single-celled organism reproducing by mitosis in the cell cycle, it's called binary fission. So mitosis is not the only thing in the cell cycle. In fact, it's a really small part of the cell cycle. So interphase is actually what takes up the majority of the cell cycle. So this whole part here is interphase. And by the way, I have this picture drawn in the classroom if you need a copy of it, but I learn best when I actually draw stuff out, so I recommend trying that too. So G1, S, and G2 are all part of interphase. Mitosis and cytokinesis is this small blue arrow here, so just a small part of the cell cycle. So let's go in order here. G1 is when cell growth occurs. So the cell physically grows, gets bigger, it makes more proteins, more enzymes, more ribosomes, more endoplasmic reticulum, everything that it's going to need to be two cells later, it makes more of all of that. So it's enough for two cells. S phase is when DNA replication occurs. So I remember this because DNA replication is also known as DNA synthesis, and S synthesis, S phase. So again, that's what happens in S phase. In G2, the cell is preparing for mitosis, preparing for division. It's making the tools that it needs for mitosis. So for example, it's going to need some more motor proteins to pull chromosomes to opposite sides. It's going to need microtubules, it's going to need actin, it's going to need centrosomes. So all of these tools that it will need to divide are made during G2. Mitosis and cytokinesis are this small little part. So they're the only part that's not in interphase. Mitosis is the lining up of the chromosomes and the dividing of those chromosomes to two sides. Cytokinesis is splitting the cytoplasm in two. So it's splitting the cells physically into two. I drew some pictures here. Those silly lights always turn it off. I drew some pictures here to kind of show the parts of interphase out a little bit more clearly, or just in a different way, I guess. So if this is our starting cell, in G1, the cell gets bigger. See how I went from smaller to bigger? I also tried to represent that there's more of the various parts of the cell in this one too. I have more blue dots. So these pink and green lines are meant to represent two chromosomes, a pink chromosome and a green chromosome. See how those are not replicated yet, but we did get a bigger cell and there's more of the other parts of the cell present. When we go through S phase, that is when those chromosomes are duplicated. So there's an exact copy made of the pink chromosome. Now there's two, an exact copy of the green chromosome made. Now there's two of those. So that was S phase that resulted in this. In G2, I said that that's when the tools for mitosis are created. So I tried to show that there are even more proteins and enzymes present in the cell, and there's these centrosomes that we'll see on the next page that are important for organizing all of the parts of mitosis. So that happens in G2. And then M, or mitosis, that is when the cell actually splits in two. And we'll look at that in a little bit more detail in a second as well. Something to keep in mind is that some cells are arrested in a phase. So, for example, our nerve cells, you may or may not know that most nerve cells, as far as we know right now, don't replicate. What you have is what you are stuck with. They don't really go through mitosis and make more and more after a certain point in your life. So they might be arrested, say, in G1 phase, so arrested or stopped. So they're not going through the cell cycle anymore constantly. They're stuck or arrested in one phase. That's a possibility. So let's talk about mitosis and the proteins involved in making it happen. You don't need to know for AP biology the different names of the phases of mitosis. You do need to know that chromosomes line up in the middle, they're pulled to either side, but you don't have to say like this is a picture of metaphase or this is a picture of anaphase. It's more about knowing how the process works. So in this picture, again, I have this drawing in the classroom if you need it to add to your notes. But I recommend trying to draw it first. I have chromosomes that are shown in green. I have microtubules that I've shown in orange. I have kinetochores, which I've shown in purple. Zoom in on this a little bit. So these are kinetochores right here and right here. So let me actually point on the board. That might be better. So here, this is a kinetochore right here, not the yellow, but the purple, and here. So there's kinetochores on either side of this yellow thing, and the yellow thing is a centromere. Okay, so there's centromere holding the two identical chromosomes together, and there's kinetochores on either side of that centromere, and we'll see what a kinetochore does in just a second. And there's also centrosomes on either side of the cell. So as I mentioned briefly before, those help organize all of the microtubules and the parts of mitosis to keep kind of those polar ends where the new cells are going to be separated and clear. Okay, so what do all of these parts do? If we were to zoom in on like this area right here where that kinetochore is attached to the chromosome, we would see that attached to that kinetochore is a motor protein that I drew in pink here, a motor protein. So that motor protein is like what we saw in the Inner Life of a Cell video, so where it kind of walks along the microtubule. This motor protein pulls the chromosome to one of the sides of the cell, and it does so by walking along a microtubule. I have an orange here. So I have written out here, in case you like to read it more, motor proteins attached to kinetochore protein and walk, I put that in parentheses because it's not like a person walking, but it's similar looking, walk down the microtubule to one of the ends. An additional protein that's involved in this process is called actin. So we've been talking about that in class. It's another protein that's part of the cytoskeleton. It's kind of like long strands. Actin is what's responsible for cytokinesis. It squeezes the cell in the middle to create those two separate cells. So it kind of is like pulling a string tighter and tighter around the cell until it's closed off and there's two cells at the end. So actin squeezes the membrane, creating two cells. And as a connection to what we've been talking about with protein synthesis, I just want to point out that actin, motor proteins, microtubules, the kinetochore, all of these proteins would be made in the cytosol or in the cytoplasm. They wouldn't go through the endoplasmic reticulum in the Golgi because they're not in a membrane, they're not in an organelle, and they're not secreted. I'll see you guys tomorrow.